Good morning, everyone. We're going to get started. My name is Moshe Lux. I'm the conference chair. If you just got here, then welcome. It's great to see you. So we started out last night with intros from myself and Peter Norvig, who gave some thoughts on the challenges of AGI, and then our first keynote speaker who talked about driverless cars and some really excellent work coming from a background of control theory and dynamic vision. And we're going to hear a philosophical talk in a little bit, and then a neuroscience talk. And one thing that sort of struck me is the sort of one of the things that's exciting about AGI is that it's pre-paradigm in the sense that there's no AGI intellectual tradition. There are lots of different intellectual traditions, and it's just very, very interesting and important that we have people from these different disciplines who are actually talking to each other and hopefully referencing each other's work and learning from each other, because that's how progress happens. And I'm going to turn it over to Dr. Ben Goertzel, who will introduce our next speaker.
Hello again. So Aaron Sloman told me he could give a one-sentence introduction of himself, which I'll see if I can remember, which basically he's a mathematician who decided that the best way to solve philosophical problems was to do AI. And I think that's an intriguing description. But to go into a little more detail, I mean, I've been following Aaron's work for a really long time, almost as long as I've been thinking about AI myself, and I think he's done a huge amount to bring together AI, philosophy of mind and mathematics, cognitive psychology, and other disciplines, sort of into a unified understanding of intelligence. Following up on the comments Moshe just made about the interdisciplinarity of the field. I mean, that takes place not only by bringing people with different backgrounds together, but by having people such as Dr. Sloman, who can bring these different disciplines together inside their own mind and their own research. So after getting his PhD in 1962, Aaron has been working on issues at the intersection of AI and philosophy ever since. His work gained something from his interactions with other early AI pioneers like Marvin Minsky, John McCarthy, and Herbert Simon, but he's certainly charted his own direction, diverging in significant ways from any of these folks. He held the chair in AI and cognitive science at the School of Computer Science at the University of Birmingham. Before that, for a long period, a chair with a similar title at the University of Sussex. He's now an honorary professor of AI and cognitive science at Birmingham. And I've enjoyed a number of his papers and presentations online over the past years. One title particularly struck me on his website and convinced me it would be a good idea to invite him to speak at an AGI conference. The title of that presentation was "A New Approach to Philosophy of Mathematics. Design a Young Explorer Able to Discover Toddler Theorems." So I like the idea of toddler theorems. Of course, that tied in with a lot of my own thinking about AGI preschool and so forth. And just looking at the young mind still in development as doing some sort of mathematics and theorem proving to understand it, practical things in the sensory motor world, I think it's a compellingly cross-disciplinary way to look at some core AI issues. So without any more introduction, then Aaron Sloman.
Thanks, Ben. Thank you very much for inviting me. This is quite an interesting challenge because I know this is a very high-powered and very diverse audience, and I'm sure everyone in the room knows more than I do about something because I spread myself very thinly. Is there anyone in either corner of the room who can hear me? Okay. And I'll try to remember to keep close to this thing, but that requires parallel processing. One minor correction to the summary that Ben reported. He left out the word failed. I'm a failed mathematician in the sense my first year was mathematics, and I was going to go on and do research, but I got seduced by philosophy, and I think it's partly because I wouldn't have been good at doing research in mathematics. I couldn't find theorems. I could find problems, as you'll see, but finding theorems is a bit different. Anyway, finding proofs, I mean. So I want to give you a brief taste. This looks dangerous. Anyway, I was using a toddler theorem about mugs of coffee and tables, and anyway, I've got a whole lot of different things which I've been trying to organize for today, and people who know me will know I never succeed. So I will go on until the time is up, and someone's going to wave a hand when I've got 5 minutes left, and then maybe 1 minute. Then I'll stop, and then we can have a discussion for a while. And as it says on here, at the bottom, if you're going to the AAAI conference, which is following on Monday afternoon, I'm doing a tutorial, which is 4 hours, if you've got a lot of stamina with a coffee break, instead of just 45 minutes or so. Now, these slides will go on my website, and they're designed to be readable on your own. So there's far too much for presentations. So you can only look at things, you need only look at things that I point at, and now I have to somehow find 2 pointers at the same time. I'm not going to try, but that's the one I can see. Turing, in his 1950 paper, which everybody quotes and very few people have actually read, said at one point in answer to one of the objections, instead of trying to produce a program to simulate an adult mind, why not rather try to produce one which simulates a child? If this was then subjected to the appropriate course of education, one could obtain the adult brain. There's a premise there about mathematics that I'm going to come to. Presumably the child's brain is something like a notebook as one buys it from the stationers. Rather little mechanism and lots of blank sheets. Well, I'm going to say exactly the opposite. Nothing like a blank sheet and lots of mechanism and all those millions of years of evolution, not just during human times, but before that with microbes and all sorts of other things. All those millions of years of evolution.
This is a moving mic if you want to walk around.
Yeah, okay.
Like a rock star.
Sorry. I'll try not to rock too much. Those years of evolution were not wasted. As John McCarthy wrote in 1996, he wrote a paper which is worth reading. It got republished about 20 years later, well, so quite a long time later, in the AI journal. It's called The Well-Designed Child. And his idea, he wrote it because it was triggered by reading the work of Elizabeth Spelke, who is one of a number of developmental psychologists who's started looking at what seems to be innate knowledge in babies. And I think there are lots of dubious things about the particular methods and the particular questions they ask and the particular answers they give, but at least they, I think, would agree with John McCarthy, and I agree with John McCarthy, who wrote, instead of building babies as Cartesian philosophers taking nothing but their sensations for granted, evolution produced babies with innate prejudices that correspond to facts about the world and babies' positions in it. Learning starts from those prejudices. What's the world like, and what are those instinctive prejudices? And somewhere else he said, oh yes, up here, evolution solved a different problem from that of starting a baby with no a priori assumptions. Now, I have no objection to people trying to build a baby robot with no a priori assumptions and see how far you get. I'm only saying, insofar as that's not my goal is to understand myself mainly, and maybe other people who are a bit like me. And I don't think that's how we work. And there is something that starts with no a priori assumptions and then builds humans. And that's a planet with a lot of chemistry and stuff, and a few billion years to mess around in, and maybe a lot of luck, I don't know. Maybe there's something inevitable about that starting state. But although I'm interested in that question, how did that work? That's not the question I'm going to talk about today. I'm really asking what did evolution do for human babies? And what would be like to do that for robots, babies. I use this to illustrate some of the sort of general theme of what I'm saying, namely evolution had many different problems at different stages in the evolutionary history of animals on the planet. Microbes of various sorts. And the last one, how many people don't recognize that, the last picture? A few. Okay, I'd better show the video. Betty was a New Caledonian crow, that one, who was brought to Oxford and was given a problem. New Caledonian crows are particularly intelligent. All corvids are intelligent. They do things you couldn't do, like build a nest by bringing one twig at a time in your mouth or just holding it with 2 fingers and somehow weaving them into a rigid structure that you can safely put your eggs in when the wind's blowing. Try it and let me know if you manage. Anyway, they do that, and they do other things as well. They also cut twigs into little hooks to get larvae out of cracks and so on. So they wanted to see if they would recognize a piece of wire bent as a hook as a solution to a problem, which is getting food out of a bucket. Apologies to all of you who've seen this before. It became very famous in 2002. The videos were all over the place. And in fact, if you give Google Betty, Crow, Hook, and Oxford, you'll get to the Oxford Ecology Lab, and you'll see lots of videos and pictures. Anyway, Betty surprised them by doing something different from what they thought. Initially, she surprised them by recognizing the hook and getting the bucket of food out of the tube, and you'll see her doing that. But then something happened that they didn't plan. Is that visible? She is there with a straight piece of wire because the male crow had taken the hook away, and she found a straight piece of wire. So she came with this piece of wire and tried for a while to get the food out, and sometimes she managed. This was trial 7. They had a whole lot of trials. And sometimes she could push the wire through the handle, press it against the opposite side, and lift it out, which is not bad for some colleagues in Birmingham are trying with children, teenagers, and they are having great difficulty with this problem if they get a piece of wire or a pipe cleaner or something. Anyway, you probably saw what happened. She stopped after a while and stuck the end of the wire into the ducting tape at the bottom and very expertly just moved around to make a hook, and then she came and used it. It's worth looking at several times and looking closely at what's going on, but I'm not going to spend a lot of time on that now. There's too much to get through. Let me show you something rather different, a baby. Now, I don't know what's going on in this baby's mind, and I don't know how the baby's father put up with what was shown in this video. The baby sitting on a carpet with a tub of yogurt and a spoon. Apologies to those of you who've seen me show it before. And this is not an animal solving a problem. I think this is an animal trying to find out what it should learn about the world by trying things out and seeing what happens. And the interesting thing is, whoops, wrong command. There's someone holding a tub of yogurt. The baby's got a spoon, and notice that he's trying to feed his leg with it. Then he feeds himself, his stomach. Then he does a little experiment. He pushed the spoon and let go. He does it again later, so you see. There he tries to transfer yogurt to the other leg and fails. Another little experiment, a better one. Then yogurt into his mouth. Now, it looks to me as if he thinks that if you put the spoon somewhere with yogurt in it, the yogurt goes to that place. And you see him trying again and failing. There he found some yogurt on the floor. It was the wrong place, so he put it in the right place on the spoon and then into the mouth. Then he tries to put more yogurt on the floor, and he hasn't understood the relationship between the bowl, the yogurt, and the floor, which has to be controlled for that transfer to succeed. And he said, I'm guessing what's going on. That's me trying to make sense of this messy, horrible demonstration. But he seems more than once to try to put yogurt somewhere on the floor, on his leg, and so on, and fails. This is aged about 11 months. I presume that not long after that, he had worked out that what you have to do is turn the spoon over. But he solves another problem, which is actually rather harder, right at the end of the video, when he decides to transfer the spoon to the other hand. And he manages. So he's got one hand holding the end, and the other hand goes, he has to somehow transfer it without dropping the spoon. And as you saw, he managed. All of these things are available on my website and more. So what's going on there? Well, I believe this is an early phase in a process I'm going to talk about, a phase which I suspect will ring lots of happy bells for people who talk about AGI as involving exploratory learning and keeping information from your sensory motor records or whatever, and then later on doing something with it. We may differ in what we think is done with it, and I'll come back to that later. So let's go back to now. Part of the point of putting that up there is that I don't think the human who's not shown in this picture starts off knowing nothing. I think there's an awful lot that is a requirement for the learning that happens later. And at a much earlier stage, of course, there's sucking. And most people think the reason why there's all that competent sucking is because the baby needs food. But I don't believe that if that were true, it would follow that you're not depriving a baby if you just give it a bottle or even stick a tube down its throat and let the food go in. I think that sucking is part of a process of exploring the world, but we can't see what's going on. It's learning. And if you watch a newborn baby or a very young baby trying to actually get a grip on a nipple, it can have a lot of trouble. And there's a lot of negotiation, and it's probably learning all sorts of things. It doesn't know it's learning it, but something is going on there. So that's an even earlier stage. Now, that depends on a huge amount of pre-programming by biological evolution. You might say, okay, we're not into that. We're doing AGI, artificial general intelligence, which is artificial as opposed to natural general intelligence. I'm not sure humans have general intelligence, but that's another issue. I want to say something about the problems of the natural case, and maybe it'll make the artificial case look a bit different. Or maybe not. We'll see. So, biological evolution had many kinds of organisms, and they had many different environments, and had many different problems to solve. And there were many transitions that were driven in evolution by things that changed in the environment and also in the organisms themselves. So maybe initially there was just a chemical soup. There may initially, or at some later stage, have been detectable gradients. If there are no gradients, then there's no point having sensors that can detect gradients or motors for moving. You just take potluck, and some survive, some not. If there are gradients, then you can go in the direction of whatever gets you more of whatever you need. But then you have a whole bunch of control problems. You have to get the information. You have to have a store of energy. You have to have effectors for putting that energy to work, and so on. If, in addition, there's some stable structures like rocks or plants that don't move around, that are not just dissolved, and you need different ones at different times, then there are new problems about finding out where those things are. You might be able to survive, and maybe some organisms do, by just being blown around and not making any use of the fact that there are these stable structures. But ones that are able to record information about the spatial layout of things and to be able to use that information in order to propel themselves around according to their needs and according to what they've learned, will be able to do things that the others can't. It doesn't mean the others get wiped out, because you always need the older, less intelligent things to be around at the bottom of a food pyramid, it seems. And the microbes are winners on every measure: biomass, number of different types, number of individuals. If you want to talk about evolutionary success, look at microbes. Anyway, so despite all their success, other things managed to find little niches into which they could evolve, and they started developing both physical structures and then information processing mechanisms to deal with the problems of controlling their own physical structures and also dealing with the physical structures in the environment. And I've given you some sort of way of thinking, which may be already familiar to many of you, which I think can lead to a collaborative project of identifying as many as possible of the changes in information processing that might have happened, because whether they did happen or not is a separate issue, that might have happened in the evolution of the planet. And that would echo work done by John Maynard Smith and Zamathuri, biologists. Have I got a slide on that? No, I haven't. Anyway, they have a book called The Major Transitions in Evolution. And to my surprise because of what I know about John Maynard Smith, information processing changes have very little role in their list of changes. And when they mention it, it seems to be only changes in modes of communication and things like developing generative grammars and so on. And there's a lot more information processing that goes on before there's communication, as I've tried to indicate, in making use of available information to get available resources to meet your needs, given your own resources for making use of information by moving around and so on. I left out some things that perhaps are worth pointing to. There are some things that have to be manipulated to be eaten, as opposed to just being absorbed through the skin. So some things you have to take apart, or whatever. There are some things that, so maybe that leads to the evolution of manipulators. But manipulators then raise new problems, which are controlling the manipulators. And there are different kinds of manipulators: claws, tongues, jaws, feet, and so on, legs, trunks, elephants, and octopus's tentacles. And they all have different, and interestingly different, problems of control. And maybe at some level there are similarities also which are not necessarily obvious. I'm told that one of the things that an octopus does is turn its apparently continuously flexible thing into a jointed thing. So it solves a problem by pretending it's got an elbow and a shoulder and a wrist or something. And one theory is that that's how arms with bone structures and so on actually evolved, but I don't know. Some of you may know more about that than I do. Okay, then there are things that try to eat you and things that try to escape. And at that stage, it's useful to start taking account of, or being able to take account of the fact that in the environment there are things that process information, as opposed to just food and obstacles and dangers and so on. And if there are things that process information, then in order to represent their ability to process information, you might succeed just by keeping track of how they move under what conditions. Or if they're getting cleverer and you want to get cleverer, you may have to have some theory about what's going on inside them, about what they can and can't see, or what they might or might not already have learned, and so on, or what they might want that they're not showing yet in their behavior. And that requires the ability to represent things that represent metasemantic competences. And I won't go into it, but there are quite a lot of problems about how that works, and even now there are debates about things like referential opacity. Who's heard of referential opacity? Not many, but a few have. Okay, well, if that comes up in question time, it has to do with the fact that if you have information about something, then the contents and the things represented are in a fairly tight relationship. If you have information about a representation of something, then that relationship is very different. And there are debates about how to deal with that. And John McCarthy, as ever, says you need a new logic or something, an extension to logic. I think you need a new architecture, but that's a detail. Okay, so these are some of the kinds of things that are related to fairly large-scale transitions. And if we look more closely, I think you'll find lots of more detailed transitions, which are interesting and worth finding out about. Some of these involve collaboration and so on. So how do the information processing requirements change across these and other cases that I haven't distinguished? How many of them map into problems of data mining in sensory motor statistics, which is one approach to trying to do AI. And it may have been what Turing had in mind when he said all you need is a blank slate and a very powerful algorithm for finding, for learning. I don't know what he meant. But I'm surprised he thought that, actually. Right, so how can we study the living things to find out what these processes, what these transitions in information processing were? And that's very hard. There's some people say, well, you then have to go and observe their measure and measure them, their movements, their behavior, their changing relationships to environments and to other organisms and so on. Or cutting them open to find out what they're made of and where the bits are, or putting them into brain scanners to find out what's going on when they see, etc. I think myself that none of these sort of traditional scientific methods will suffice. Mainly because in order to get good answers to these questions, you have to develop new thinking tools. You have to develop concepts. You have to develop new forms of representation, as has happened in the history of AI and long before that in science. For instance, when arithmetic was represented using the Arabic notation instead of the Roman notation, that made a huge difference. When zero was added to arithmetical concepts, that made a huge difference. When Newton invented calculus, building on top of work of other people, and Newton and Leibniz, I don't want to offend anybody, invented calculus, that made a huge difference. A lot of Newtonian mechanics and stuff that came after would not have been possible. And in the last 50, 60 years, we've been learning all kinds of things about information processing. And most of that work has come not from trying to study natural information processing systems, although that has contributed, even from the early days. John von Neumann and others were trying to think about how you build things that work like brains. But a lot of it came from people just trying to work like engineers and say, how can I make things do so-and-so? And they had ideas and they tried them out and they failed. And then they studied how they failed and that gave clues as to what to add. And that, I think, is a very powerful way of driving the development of new ideas for expressing new theories, which make you then go and look at the old problems in a new way. And that's one of the reasons why, with some colleagues in biology and AI and so on, we're trying to promote a new subfield, which instead of being biologically inspired AI, is AI-inspired biology. You go and ask questions about organisms that you would ask if you were a robot designer trying to make something that could do that. And those questions are not necessarily the same questions as you ask if you're a biologist or neuroscientist or whatever, asking how to do it. Anyway, so I'm suggesting we need a rather less direct approach than the standard scientific method, and that's because studying information processing systems is somewhat different from studying chemistry and physics and geology and so on. And I'm not going to elaborate that now, but I may come back to some aspects of the difference later. Well, I've said all this already. You start learning to ask questions like, what ontology does that system need? What forms of representation? What kind of information processing architecture? Questions like that were not needed by Newton or Einstein. Or Darwin needed them, but he didn't know he needed them. And as a result, he wasn't able to answer some of the questions he was thinking about. And neither were his supporters. I think now we can go and start trying to defend Darwin against some of the critics who said his evidence only showed that perhaps physical forms and physical behaviors evolved, but you couldn't have got consciousness, mental states and processes coming out of natural selection. You can't show transitions in the fossil records of the sort that would be required. And there was a lot of bumbling, but even one of his great supporters, T.H. Huxley, used the phrase that is now bandied about. Many people think it was invented recently. I won't name the alleged inventor. The phrase, the explanatory gap, some of you may have heard about. That was a hot topic in the mid-19th century, and there's a lot of stuff, a lot of debates that went on then, which were sort of reinvented or rediscovered as new terminology in the last 10 or 20 years, but it's the same old stuff. And Darwin and most of the current philosophers thinking about these things do not have the conceptual tools for coming up with deep theories. And I think we're in a much better position after having developed the conceptual tools needed to design working systems of the sort that Darwin claimed evolved, but he didn't have the right conceptual tools to describe what evolved or what sorts of mechanisms might have had to change to make that evolution possible. Okay, so that's part of the reason for all this mixing up of disciplines that Ben mentioned earlier that I engage in and confuse myself and everybody else in the process sometimes, I think. I now want to go back to this thing about the tabula rasa, the problems of learning about the environment. This is part of a website where I posed some questions for some people who told me they could develop general purpose learning systems by doing dimensionality reduction. I said, okay, try this. And the idea was if you've got a pixel array and you have things like what I've shown you there, and the things move around, and the task is to be able to predict what's going to happen next, can you do it? And there are interesting questions about what you can and can't do. And in some cases, if you just have something like that and it moves horizontally, or maybe if it rotates, if it moves in a way that can be expressed as a simple change in a few variables, then probably one of the many approaches that people in this room know about would work very well. At the bottom it says show the rotator, so I'll show the rotator. The rotator is here. That's a rotator. Now, if your pixel array has got stuff like that in, and you want to be able to predict what's going to be happening next on the basis of what's been happening recently, if you try to do it using just an ontology that is based on the contents and the changing contents of the image, then it's going to be difficult, I think. Now, there may be people in this room who say, oh, I've got an algorithm that'll do that, no trouble, and then I'll learn something. But my guess is that what you really need is to know that there's more than 2 dimensions, and to see that thing as a cube that is opaque, has a number of surfaces that can be visible but not all at the same time, and it's rotating. And maybe a learner can benefit from being able to control that, and perhaps that might help. I'm just using a mouse here. This is a nice gadget on the web in Java. You could also have a Necker cube, which, when we look at it, is rarely just seen as a 2-dimensional pattern. We will see it as a 3-dimensional pattern, but typically we can see the same data as 2 different cubes, where the differences involve geometric differences like different faces are close or far, and lines are sloping up or sloping down, and so on. And again, trying to explain what's going on there, one needs to have an ontology that goes beyond the data. Let me just stop this. Well, go back. So what I would like to propose as a collection of experiments is that we produce a collection of videos generated in a variety of ways from structures of various kinds, and then see what the problems are in having a totally knowledge-free but powerful learner coming up with good explanations which enable predictions to be done and interpolations if you missed a bit of it, to say, oh, in between you this and this, it must have been there, and so on. Do any of you know Johansson's pictures? He took photographs, Gunnar Johansson, photographs of people with lights attached to their joints, videos in the dark, not photographs, sorry. And they were doing various things like dancing, climbing up and down ladders, doing press-ups. And humans looking at those instantly see not just 3D structures, but you can actually see the effort going into the push-ups and so on. So there the ontology goes even beyond 3D structures. You use your knowledge of human beings, and you use sort of biological concepts like effort and so on in interpreting them. And so the question is, where could that come from if it was to be learned just from the data and maybe interacting with the data? I'm saying that probably a lot of it comes from that evolutionary history in our case, but maybe there are other ways of doing it in artificial systems, long direction. While I'm here, let me show you another toddler, slightly older toddler. Wrong one. This is a succession of snapshots from a video. The video is too short and goes too fast, so I should find a way to enlarge it, but if I do that now, it'll take too long. There's, I think about 18 months, I can't remember, child holding a broom. And you can't see it, but the back end of the broom is stuck between a couple of vertical railings. He's on a landing. He tries to move it, and you don't see that, and move the thing sideways, and he can't. So he looks back and quickly realizes what he has to do. He pulls the thing forward, and then a whole bunch of other things happen. Looks around, moves the thing forward, So he knows the theorem about how to move something that can't be moved laterally, but is in a situation where it can be moved at 90 degrees to that lateral motion, which will then enable it to move laterally if you move it out. He doesn't know he knows it, he can't state it, but he clearly, instantly did what he had to do. Then he carries on doing a few other little things, transfers it to the other hand, and pushes forward. And then he's stuck. Can't push it further because the broom hits the skirting board. So he pulls it back and aims it in a different direction. Goes down the corridor. And there he's not only pushing the broom, he's maintaining his balance and controlling also dynamical systems to keep him moving. An interesting thing happens near the end. There's a door on the right of the corridor. Whoops, I'm sorry, this probably gone. I should perhaps just stick with the video. These problems wouldn't find some technology for slowing down the video. He notices a door approaching. So before he gets to the door, he swivels the broom handle round to the left so that when he gets to the door, the broom is in the right plane to be pushed straight through the doorway. And he goes through, and then the video ended shortly after. I haven't had this problem of the thing moving down in the past. I should get another, I should use a different viewer. Anyway, so what I'm suggesting is that by that age, he had clearly learned quite a lot of stuff about how to move an object, in this case a long, thin object, that he could hold at one end and push along the floor, so as to produce behaviors. And he wasn't always simply doing online control of motion in order to modify the current sensor values. He was anticipating. He noticed a possibility and acted on it. And there's a huge amount of work on robotics, which produces very impressive robots that can juggle, that can balance a broom. I think there's a reference to one last night. That can catch balls, that can move through doorways before they shut, and so on. But I don't think any of them know what they've done after they've done it, know what they didn't do, know why they've done it, nor, I suspect, do they, like this child, know what they're going to do a few steps ahead and why they're going to do it, which I suspect he did. Whereas the toddler who was transferring a spoon from one end to the other probably didn't know he was going to do that until almost immediately the opportunity arose. But once it started, I think in some sense the goal was already there, driving the selection of motions of the hand to enable the thing to be transferred. That was about 11 months, I think. So part of my story is there's a huge amount of fascinating data in babies and toddlers, and I've just chosen a few things which were buried in lots of videos I was shown. And I'm trying to get colleagues and friends and other people to do this with their babies. And the trouble is it requires you to set yourself the discipline of taking 30 seconds every week or so, or more or less or whatever. Deb Roy, of course, has a major project of that sort. But I think in some sense he may have more data than anything can handle. But I don't know what the current state of that project is. Frank Gurn in Aberdeen has been acting in this way, and have I got his? He sent me some stuff last night, and here, if I can make this run, it's his daughter. And he set up some apparatus for her, and she has obviously learned some toddler theorems. But she has some problems. She's seen something else that's out of reach, and she's trying and getting into difficulties. She tries something else, like to get the keys, but she won't do that, and somehow manages. But notice she uses the other hand to help. Can't bring it to her. Realizes that the tool can bring. Yeah. I can't remember, but I would say under 2 years. I've got an email message which has the email me, and I'll tell you. There's a huge amount of work in psychology on trying to find out what babies can do and when they can do it, and whether if you change the situation slightly, they can do it a month earlier or a week earlier, but without any attempt to find out how they do it and what the problems are that have to be solved for them to do it. So I don't know if that was part of the background to that question. From my point of view, at any stage that it happens, there's a question: how was it done? What was going on? And at the moment, I think we know very little. But if there's time, I'll say a little bit about the theories of Annette Komoloff-Smith, who was one of the people who worked with Piaget and then developed her own line of thinking, which in some ways is very different from Piaget's. And I only realized how close it was to what I was talking about very recently. Okay, how are we doing for time? I think I'm getting quite close to running out of time. Let me give you a little exercise. You may have noticed that if you have a plastic shampoo container, then if you turn it over and squeeze, when it's new, it's very easy. You just squeeze. If you've had it for some time, it's better to wait a little while before you squeeze. Has anyone noticed that? Question is, why? Now, part of the point about that exercise is that the explanation is not in anything you see. You have to know something about substances, and in particular about shampoo, namely that it's viscous, it flows down, and if you squeeze too soon, it won't have reached the nozzle, and all you'll be getting when you squeeze is air until the stuff has got to the end. And I believe that the ability to learn about and reason about stuff, different kinds of stuff, clothing, skin, fur, teeth, rubber, plastic nowadays, wire, and so on. Wire can have a lot in common with string and also be very different in the details, is a crucial and substantial subset of what toddlers learn. And that's part of what I think is going on with chewing and sucking and eating as well, not just getting food. I don't know of people working on robots that are learning about kinds of stuff, although we have a PhD student in Birmingham who's getting a robot to understand what happens with sponge when you press it and so on. Maybe other people are doing that too, and email me if you know some interesting work on that. I'll give you another puzzle. You may have noticed I have an ink spot here. And when I found that last night, I was very annoyed because I was sure I had trained myself always to retract the ball before I put the pen in my pocket. And when I found this, I thought, this is just, it doesn't make any sense. I've trained myself. And then I had another idea. And I'm going to leave it to you to think about what the other idea might have been, and maybe come back to that. Is it obvious to anyone what the other idea might have been? Oh, you have to know I flew here yesterday. Maybe that makes it too obvious. Okay. So what some of these last few points are trying to draw attention to is the difference between somatic ontologies and exosomatic ontologies. A somatic ontology is something that is either that refers to the contents of central data and motor signals, or any mathematical relations that you can compute from those. So the soma is the body, and the somatic ontology refers to things going on inside the body. An exosomatic ontology refers to things going on outside the body, and maybe even out of sight, like the shampoo in the squeezy bottle, or the insides of stuff that behaves differently from other stuff, that makes the stuff behave differently, or the hidden sides of a rotating cube. So there's or even the visible sides of a rotating cube. When you see that as a thing that's in space outside you and rotating, you're using an exosomatic ontology. So there are some people who try to start with nothing but a somatic ontology and powerful algorithms and do everything. And one of the interesting questions is, can you get from that in a sensible way, in a reasonable time, to an exosomatic ontology? Now, there is a way where you can obviously do it if you've got a formalism for expressing all possible theories, and the formalism gives you recursively enumerable theories. You just try the theories one by one, and all the data you've got, thanks. And until you find one that works well. The trouble is, maybe not even a planet is going to be, and 4,000 million years will be good enough for some of the problems. But I don't know. Maybe I haven't just understood the combinatorics. But I don't think that's what young humans have, and I suspect that well-designed robots will not have that either. They will have some precursors of an exosomatic ontology or some explicit exosomatic ontology, like the notion that there are things that exist independently of whether they're being perceived or not. And those things may be of many kinds, not just things like balls and cups and so on, the things we often refer to as objects, which are being studied quite a lot in developmental psychology, but things like surface fragments, which can have orientation, curvature, reflectance, and other kinds of properties. Processes of various sorts, like becoming more or less curved, or stretching, or moving in various directions, or rotating. These are things that can happen to things outside you, and you may see them happening. And if you have the right sort of ontology, then you can describe them. And, importantly, if it's a degenerative form of representation, you can combine them in ways that you've never encountered to make predictions, to imagine things, to set up goals. And I think that that is part of the apparatus that drives the explorations of very young children in ways that initially is highly, I think, determined by the genome reacting to what's in the environment. So if you see something happen, then you can get a motive to try to make it happen again, or to make it happen in reverse, or to make it happen with variations. And if you watch children a lot, you see a lot of that going on. They do things and they try, and sometimes they'll do something totally unrelated to what they were doing before because they've just noticed the possibility. Not because of any reward or anything. I think they're built to do that because they'll learn things. And then a lot of behavioral competences develop, which are shown in various skills, like walking and climbing and feeding yourself and so on, pushing brooms around corners. But something else happens as well, and this is what Annette Komolov-Smith talks about in her book, Beyond Modularity, published in 1992. The details will be on this. I've actually started to produce a little website about her book. And whereas Piaget had said that children go through stages, and in the first stage, sensory motor stage, or whatever, I can't remember all the orders, they're pre-operational, operational, and other kinds of stages, with fuzzy transitions between them, but what you can do varies from one stage to another, and they're roughly age correlated, but there are individual differences and so on. What she says is, those stages are roughly applicable not to ages of development, but to stages in learning about a new domain. So if I had time, I could introduce you to a new domain. For instance, a domain of shapes you can make with a rubber band and a collection of pins on a pin board. And you can play and do things and make the letter T and capital H and so on. And then I'll ask you to make a capital outline A, and you may discover that it's impossible. And then I might ask you questions about what's the minimum number of pins you need to make, say, a figure that's alternating convex and concave vertices. So you can play and you make discoveries, but something happens after a while. What you've learned empirically gets reorganized. You can do lots of things, you have behavioral competence, but Komolov-Smith talks about representational redescription. And I think what she's saying is that the information gets put into a new form, which is more like, she doesn't say this, more like an axiomatic theory, but I don't think that necessarily means it's using any branch of formal logic that we know about, maybe something else. We know one case of that that's very familiar, language learning. Children learn to speak and understand things pretty well at first, and then something happens where they start saying things like, Johnny hitted me, and he ran home, and he sitted on the chair, whereas previously they'd say, Johnny hit me, and he ran home, and he sat in the chair. That transition suggests that what's going on is that previously the children had learned a lot of things at work, they had the behavioral competence, but then they reorganize that. It's a case of what she calls representational redescription. And if you think about it, you'll realize that enabling a generative system based on a syntax to deal with exceptions is a non-trivial software engineering task, to add the ability to handle exceptions in the inputs and the outputs. But kids solve that problem somehow. They don't know they're doing any of this, by the way. There's no claim that there's self-knowledge. That's a later stage. And what I'm suggesting, and it turned out I discovered Commonwealth Smith, for quite different reasons, based on her empirical observations, was also suggesting this, but we were using different language. I was talking about kids discovering toddler theorems and learning things empirically and then reorganizing that knowledge so they could then deal with novel cases. She was talking about another way, which she calls representational redescription. And so I think that work needs to be brought together, but we're both saying it's not just language. And I'm suggesting that there's a lot to do with things like learning topological relationships, learning geometrical structures and processes, and learning a lot about processes and the conditions under which they occur and the constraints on those processes. So you can learn a lot of it empirically and learn to do things and learn to predict what's going to happen, but then you can transform that knowledge into a deeper form. As she points out, this also happens at different stages of development, and it can also happen in a culture. So, for example, there was probably a lot of knowledge of geometry before some ancient, whoever it was, Egyptians or whatever, organized that knowledge into what we now think of as Euclidean geometry. And then still later, that organization was itself reorganized, saying that it could be expressed in terms of a formal system, predicate calculus of one sort or another, by Hilbert and other people. And I'm still playing with ways of trying to do that by eliminating the parallel axiom, but still talking about rigid motion. And it may be impossible, but I want to see if I can, because I suspect that would fit some of what goes on with youngsters learning about space and time, because I don't think they have the parallel axiom. It doesn't come into their experience, and they don't need to know anything about it, but they do need to know a lot about rigid motions and so on, and things preserving length as they move. Okay, so what I'm saying here is summarizing what I've just said at greater length. We need some mechanism which is good at generating exosomatic ontologies, and I suspect it needs to have a lot of prejudices built in. But those prejudices would not be, this is the ontology you need, but this is a way to find out what ontology you need, given some very abstract assumptions, which I presume include you're in a three-dimensional world. You're in a world in which there are structures, some of which change, some of which are preserved. You're a world in which the structures are made up of kinds of stuff which have different kinds of properties. Not that they're rigid, solid, elastic, or whatever, but different kinds of stuff. You find out what they are by playing. And maybe you can find out what the differences are by finding ways in which they respond to what you do to them and develop a theory. And I suspect a lot of that's going on. I suspect nobody knows what's going on of that sort, but it's happening anyway. And I suspect it has the stages that Carmel Smith has been talking about, which involves having empirical learning followed by reorganization, which enables things to be worked out instead of being learned empirically. There's a very good book by Diana Pick and Eleanor Gibson, the wife of James Gibson, on child development. I can't remember the title. But they talk about the kinds of ways children can learn something. They talk about learning by experiment, learning by observation, learning by imitation, learning by being taught, and they miss out the crucial thing that I think is what makes us human, namely learning by working things out for the first time. And that goes back to the bug in what I think Turing was proposing, because he talks about teachers having to be there to train this robot that starts with an empty notebook. Where did the first teachers come from? If you need a teacher to learn mathematics, then the process couldn't get started. So what I'm suggesting is that there's something about the biological process of giving animals better and better abilities to deal with the problems they faced in coping with complexities, both in their own structure and motion and in the environment and their competitors and so on, which somehow enabled evolution to produce systems that could do that representational redescription or axiomatization or theory construction or whatever, so that things that initially were learned become theorems in a generative system, which can go beyond the sort of thing that fits naturally within many statistical models and neural models, which can only cope with an envelope defined by the data, but have what Chomsky called infinite, the other kind have infinite generative capabilities of a different sort. you can have longer and longer sentences with more and more nested subclauses and all sorts of daft people inserting things which you can't predict and you've never heard before and you'll never hear again, but you understand them. And I could go on for another 5 minutes with this stupid sentence just to illustrate that point. But the same thing applies with Heath Robinson, structural apparatus and machinery and so on. Okay, so that's the observation. That's what needs to be explained. And I really look forward to learning how it can be done. The task's too difficult for me, but I think a team of very bright people working with a collection of disciplines might be able to make some progress. Thank you. Has anyone worked out the solution to my ink spot that occurred to me after I cursed myself? Yes?
You said you flew on a plane and the pressure changed.
Yeah. Okay, so have you ever been faced with that problem before? No. So you're doing what I was talking about. You had some generative knowledge you were able to apply to a novel situation within a few minutes. You didn't have to search. Okay, thank you.
We have about 5 minutes for questions. We'll do things a little differently today. If people want to line up there and there, I will give them microphones so everyone can hear them and things will be recorded.
And I have a problem with it. I'll do this one on the other end. There are lots of interesting problems about knots and tangles.
Oh, yes, fascinating. Let's test my toddler theories.
Yes, thank you.
There you go.
I would like to give an argument to support the Turing point. If you take genome, it's about a billion bits in it, and this is all information that is transferred to INNET. And according to current knowledge, about 90 or more than 90% of the genome is just redundancies. So you have like 10 to 7 or 10 to 8 bits transferred to INNET, and of this there are many information about organs or immunological systems. So you are only, the crucial information for the mind is about only a million bits, 10 to 6 bits. So we are just a million bits away to making mind, because there's no other information transfer. And so I think that like Intel processor is about 100,000 bits complicated in terms of complexity. So we are only just this away from making it. And even if you take point of view of Penrose, that is quantum machinery, anyway, the information transfer is only limited to that. So, and if you think that Turing machine is not enough, I would say that according to Turing thesis, you can simulate any machinery on Turing machines. So it doesn't matter whether it is a computer. Sorry. Yeah. But the point is also that you have examples of people who are disabled since birth and who are blind since birth, and we find them intelligent. So all these arguments. What do you think about this argument against? To support Turing point?
Well, you raised a lot of points. I don't believe that everything on our planet can be simulated on a Turing machine, including 2 Turing machines controlled by a Geiger counter, which determines which one runs and produces a 1 or a 0. But never mind that. Yes, the people who are born disabled without limbs learn a huge amount. The one thing they can't do is learn it by doing the same kind of sensory motor exploration that other organisms do. So that raises deep questions about how someone like Alison Lappo, who's a famous limbless artist, and if you look up that name on Google, you'll learn about her. As an adult, she's clearly got a deep understanding of the problems her child has. She's got a normal child. She had a genetic defect. So yes, there are all kinds of things that are learned, and we don't know how. I would be quite happy to wait and see you and others develop the viewpoint that you're presenting. I don't have knockdown arguments saying it's impossible. I'll just add one little thing, that the genome is not the only thing that controls the development of an organism. Of course, the mother's provides an environment which in itself is a product of the genome, but not just the genome, the genome and the chemistry of the world. So we don't know what's going on there. But you could say that just supports Turing's point. We would have to talk more about that in detail. I don't think anything obvious follows from those points.
Yes. My own work and perhaps other projects, other cognitive architectures here have the approach that that goes back to the paragraph that you showed from Turing's paper precisely, where I'd like to create a student system that's capable of being taught. And I'd like to drill down into your criticism of that approach. One criticism I picked up was that you believe that the students should be able to work things out by themselves. And I ask, is that itself a skill that could be taught by a mentor?
I think it comes in a way that I've been discussing with a biologist, Jackie Chappell. We presented a paper at AGI in 2005, and then a couple of years later, a paper in a strange journal called the Journal of Unconventional Computing. We were invited to expand our AGI paper. And we have this notion that the genome can produce behaviors in an environment via different routes. And in the simplest systems, there's just genome produces physical structures which produces behaviors like reflexes and so on. And then in slightly more complicated things, you have a genome produces physical structure which encodes competences which are generic. They may have conditional forms, so what you do will depend on some of your internal states and goals and things. At a later stage, partly as a result of what goes on over time, feeding back into the system, you have things which are meta-competences, which are partly determined by the genome and partly by what's been learned in the earlier stages. And those meta-competences can operate on previous competences and generate new competences and so on. And they, in turn, may sometimes generate new meta-competences. So I feel, and certainly my experience is, that I've gone on learning to learn throughout my life. And it's clearly true that when kids come to university, there are things they cannot learn that they, for instance, they're doing a degree in physics. By the time they're in the final year, they can learn things that they couldn't learn earlier. So yes, they're being taught, but they're not being taught by somebody saying this is how you do it or whatever. There's an interaction with a very complicated process, which I don't think we understand. But that's a very crude and unsatisfactory schematic way of suggesting what it might be like. But the idea is that at every stage, this is the wrong way, the inference of the genome is there, but how much of it is from the environment can vary depending on which part of the system it is. And in some cases, it may just come from internal explorations, which were just lucky. You just happen to get an idea.
I just want to comment a little bit about the chicken and the egg question, right? This principle side, the Turing question. Now, yesterday I made a slight point that language, for example, could not have. It's implausible that it would have developed like this out of one mutation because the teacher is, children that grow up in the forest, et cetera, et cetera. So you need that. But between the one aspect of the autonomous agent that can learn anything and the agent that needs to be taught, wouldn't evolution more plausibly have been a process of co-evolution in which parent and child evolve together to solve difficult problems that the environment poses, without which there wouldn't be even a need for all this intelligence?
Well, not just parent and child, but social systems and individuals and child. I would just like to take a minute to offer a different view of language learning. Do you know about the deaf children in Nicaragua? How many people know about the deaf children in Nicaragua? Not very many, but some do. Okay, you'll probably know what I'm going to say. Crude, very interesting story. I'm going to give you a crude, very short version of it. If children are born deaf or become deaf very early on, it's very important for them to learn a sign language as opposed to being taught to lip read. There's a lot of evidence that if you try to make them just lip read, which in some cultures was thought the right way to do it, they're seriously cognitively deprived. Whereas if you let them learn a sign language, then they get great benefit from that. So in Nicaragua, sometime like 30 years ago or so, I forget exactly when, it was decided that because there's a great shortage of teachers of sign language, they would set up a place somewhere in a coastal town and bring the teachers and the deaf children from all over the country to this place. And then something happened after the children had been taught by the teachers, who themselves had not had the benefit of that early training, but they had learned sign language and they could teach it. The kids suddenly started doing things the teachers couldn't understand. They were signing away and apparently having fun and laughing and whatever. So they had to bring in linguists and anthropologists from other places who worked out that what the children had done was create a language of their own, which started, it was sort of bootstrapped by what they were being taught, but they did something else. And I think, partly inspired by that, but also other observations about language learning in children, that the notion that language learning is a sort of data mining is totally wrong. That you should think of language learning as being a process whereby an intelligent young toddler who has lots of things to communicate but can't communicate tries to find ways to do it and does so in collaboration with the other people that is going to communicate with. And if they are deaf, they'll do it through signing. Maybe even if they're not deaf, they'll do it partly through signing. And they will, because of that problem-solving mechanism having certain capabilities which are genetic, they will come up with something rich and powerful, as happened there. And so I think that what's going on is that children who appear to be trying to find out the way adults talk are actually trying to find out how to talk, and they are in a minority, and they're trying to create a solution to a problem that other people have already solved. So they're forced to accept most of the majority solution, but they often have their own little quirks, and among twins, you find that happening.
I don't want to. I know about the study, and I would never want to belittle the creativity and the spirit of young deaf children in Nicaragua. But that is the conclusions of that study have been much discussed, and that the actual way to find out whether that means spontaneous creation of language, it's an unethical experiment, right? Because they do get sign language even when they don't get sign language teachers, because that's how they communicate with their caretakers.
I agree. It's difficult to do, and I think the only thing is to get a good theory which fits lots of other phenomena as well as those. And part of the theory will be what's going on, to explain what's going on before children learn to talk, when they're doing a lot of problem solving and communicating. They'll go and turn the mother's face to look in a direction. So they have rich and complex motives, thoughts, and then later they learn to communicate. So they must have had an internal language all along. Sorry, I'm just taking my power supply. I'd forgotten where it went to. There we go.
Right. So we have to move right along. And the next keynote is going to be a bit of a taste of the neuro session that's coming up later today. It's my pleasure to introduce Ed Boyden. He's professor of biological engineering and brain and cognitive sciences at the MIT Media Lab, the MIT McGovern Institute, and he runs probably one of the largest, maybe the largest MIT Media Lab that's there, about 5 times the regular size. I think one of his former students is actually here. I'm not sure if he's here yet, but is David around? Well, anyway. So Ed co-invented the technique called optogenetics, which I'm not going to talk about. Otherwise, I'd probably be kind of giving you a spoiler. But it's definitely one of a range of very fascinating new tools for controlling and observing dynamic networks in the brain. I'm not going to read too many of his accolades, but anyway, so Ed has been named one of the top 35 innovators under the age of 35 by Technology Review in 2006, one of the top 20 brains under age 40 by Discover magazine. He received a Paul Allen Distinguished Investigator Award, and of course he spoke about his optogenetics invention on a TED Talk, and there's so much more. I just can't list it all. Anyway, please welcome Ed Boyden.
All right, well, thanks so much. I guess what we're going to try and explore today is a little taste of how we're trying to devise technologies to help us understand more about how the brain computes. And one of our working hypotheses is that we need to develop the tool set that allows us to get to the heart of some of these questions if we want to be able to confront the deep questions of how brain circuits actually perform computations, where we can leave the word computation sort of undefined for a second. And part of the issue, I think, with the brain is that it's incredibly complicated. It's made out of thousands, if not more, kinds of different kinds of circuit element that are heterogeneous. And even knowing what level of abstraction one has to think of the brain at is difficult. Is the cell the right level? Are there subcellular compartments the right level? And there are people, of course, who focus upon individual molecules, and then there are those who focus upon imaging the brain using non-invasive strategies. That's interesting. It always happens when you start talking about solving the brain that your computer starts to flake out for the irony effect. So let's try this. Now I see the problem. There's a mismatch in the resolution, so it's recomputing it twice or something. Okay, so the basic issue is that the circuits of the brain are extremely heterogeneous, and right now one of the ideas that's come to dominate is how do we start to understand that heterogeneity? How do we understand the building blocks of the brain? Because if we don't understand the building blocks and what they do, then it's very difficult to go beyond that and start to see how they work together as circuits. So for example, there are small inhibitory cells, there are different kinds of cell that do different things in the brain, and in order to tell how they work together, ideally we would be able to record from and perturb these different cellular elements. Now, of course, this is a working hypothesis. It's quite possible that the circuit elements are not cells, and that this is an approximation for the time being of what we are doing in order to try and understand things. And up front, it's not even clear how many circuit elements there are. And so, if we were doing this in an incremental way, we'd like to know the exact list. We'd like to know exactly how they work together. We'd like to know what are the properties of these elements that are most important, because obviously some of their attributes might be irrelevant in terms of actually understanding their function, and some of them might be very important, but we don't know yet how to encapsulate those properties in a meaningful way. Then, of course, you can get to the point of trying to understand how they work together. Data, of course, has been kind of a theme of the conference, is not always enough. You also have to have a way to understand that data. And so really what you need, I think, are tools to help you classify and characterize these building blocks at different levels of abstraction. You also have to be able to observe and watch them, because without a basic data set of that kind, it's very difficult to know what they're really doing in terms of their temporal interaction and how they work together. And then finally, ideally you could causally test these things by controlling them in order to delete things and drive information into certain cells or other elements in order to understand how they work. So I'll tell you the story kind of in reverse order, where a lot of the talk will be about 3, and then there'll be a little bit about 2, and then there'll be very little about 1. And that's partly because I think of the fact that there have been a lot of hypotheses in neuroscience where causality had been one of the lacking elements, and being able to delete or drive information into different elements is helping us actually clarify which level of abstraction we need to worry about. Is it the cell? Is it a subcellular compartment? And so on and so forth. So there's a bit of a loop here where I think the logic is sort of circling about. It's not a linear, we begin with this thing and we're going to keep going until we get to the end and then we're done. So we'll start with control. And I think control is a useful thing to think about, because there's 2 really good reasons why you can use it to solve problems. One is that you can actually assess the causal role of an element in a computation or behavior or pathology. And given so many different cell types in the brain, which have been observed in various incarnations over the last century or more, being able to figure out what it's actually doing by deleting it just for a second or by driving it can be of help. But the second reason is also one that's of great interest, and that's because we want to understand the brain not only for the philosophical and computational reasons, but also, brain disorders are a big issue. They are largely incurable. Many of them are untreatable even. And for disorders like Alzheimer's and stroke and various forms of deafness and blindness, there's very little that can be done. So we really set out to try and start with control, both to try and figure out how to start teasing apart which of these circuit elements do what, but also to try and figure out if we could do some of this fixing up of things. So given that the animations aren't really showing, I wonder if this will be going to work out quite well, and maybe it won't. So maybe I'll skip to the traditional slides just in case. What's that? Well, the other one, okay, we can give it a try. So this is kind of where we tried to find ways of controlling neural circuits. This is a species of green algae. And algae, like many organisms, have to sense light in order to metabolize and photosynthesize optimally. And neurons are electrical devices. So it turns out that algae sense light by converting light into electrical signals in a little organelle called an eye spot, which acts a little bit like our eye. And by taking the molecules that sit in the membrane of this eye spot, which you can think of as a little solar cell, when light shines on them, they'll translocate charged particles from one side of this membrane to the other. given that neurons are also electrical devices, if we put these molecules into neurons, then shining light on those neurons should be able to change their electrical potential, and therefore, depending upon which way these charged particles are going, you should be able to turn these cells on or off. And so we're fortunate because it turns out that this molecule, and many molecules like it, is completely encoded for by a small gene, only about 900 DNA bases. Encodes for this incredible, light sensor that will translocate charge. And so we and many others now have taken molecules that are viruses or other methods for delivering this gene into neurons, and in order to make specific neurons embedded within a dense neural circuit sensitive to being optically manipulated. So, for example, this opsin here, which comes from the green algae, when it expresses into the cell membrane, when light shines on those molecules, they will open up a little pore, translocate positive charge into the cell, and positive charge going into the cell will activate it. And so, as you might guess, this is now, assuming an abstraction layer that we can operate at. If we falsify that abstraction layer, and that's to some degree already happening as people start to look at how subcellular compartments can drive activity that manipulates other parts of a subcellular compartment network, then we can try to sort of bootstrap our way into developing new tools that help us navigate into these neural networks. So I should point out that a lot of luck was involved in getting this to work. There was no reason this algal gene had to even work at all in neurons. Over the last couple of years, this started with a collaboration with several investigators, Georg Nagel and Ernst Blomberg in Germany, and also Karl Deisroth at Stanford. And we published a paper a couple of years ago where we showed that you could take this algal protein, put it into specific neurons using a virus, and then you could then activate those cells with blue light. Since then, we and others have found a whole series of other molecules, including light-driven pumps that can pump negative charge into cells so that we can turn them off. And these tools are now in use by many hundreds of groups around the world to try and drive information into or delete it from specific cells. Now, one of the big issues is getting specificity in the cells. There are many kinds of cell in these network. And one of the things that we've had to do, and now there are probably as many ways of doing this as there are methods of getting genes into cells at all, is to get these genes into specific cells within a network. So for example, you can imagine that you can take a virus that only will bind to or be taken up by some cells and not others, and if you can get those viruses into a piece of tissue, then just some cells in the network will become photosensitized and the others won't. And there are many other ways of doing it as well. You can use promoters, DNA regulatory elements that you can place upstream of the opsin gene, the light-activated protein gene, and now those cells will become photoactivated, but their neighbors will not be. So there's different things you can do with this. I'll tell you a couple short stories about what you can do with this. One of the things is to try and figure out how the brain makes decisions. And to be upfront, this kind of thing is happening at a very early stage now, but I thought I would tell you one short story, which is a collaboration with Christopher Fiorillo's group. And one of the questions is, what are the neurons in the brain that can make an animal do more of whatever it was just doing? And we've, I think, all heard of stories and scientific results such as where rats will press a lever in order to self-stimulate. This is driving an electrical current into the base of the brain in a certain area, and that's then causing the animal to feel either some kind of pleasure or compulsion or other effect that causes them to do more of what they were just doing, in this case pressing a lever. And so one of the questions is, can you pinpoint the exact circuit elements that are actually involved? The electricity that's used in a classical sense will drive all of the neurons in a volume, but as we just pointed out, that volume contains thousands of different kinds of cell. And so we decided to try and test this out using a viral method of targeting dopamine neurons, which are one of the major candidates thought to be implicated in this process. And the very simple experiment was to take a box and to use a mouse, which is very common in neuroscience. And there are 2 points in the box where things happen or don't happen. If the mouse goes here, he gets a pulse of blue light. And if the mouse goes here, then nothing happens. And so if indeed we are driving neurons that reinforce whatever was just happening, we can then we should see the animal go to the blue light driving spot over and over again in order to earn these rewards. And so we use a strategy to target these cells using a combination of transgenic mice as well as viruses that allow us to selectively label these dopamine neurons. And there are about 100 different such mice out there. So it's quite simple now to actually deliver viruses into these parts of the brain to make them photosensitive or to make specific cells within that part of the brain photosensitive. And so this allows us to then to try and assess what these different cells do. So these are the dopamine neurons and deep in the brainstem. And as you can see, every time the mouse goes here, he gets a pulse of light. There's an optical fiber beaming light into the brain. And the mouse will go here over and over again, working for that little pulse of light. And so that's interesting because now we've actually pinpointed a specific class of cell embedded within this very dense region of the brainstem where so much is trafficking back and forth in terms of information as being able to reinforce whatever the animal is just doing. So a very interesting next question, and we'll talk about this towards the end of the talk, is to try and record throughout the circuits that are receiving information from this cell class. What's actually happening in a circuit to cause the decision to do something again, or to choose to go to one point rather than the other, sort of the fundamental mouse decision-making kind of task. So one of the big issues that we had to confront is to figure out how to get light into the brain, since the brain is obviously not transparent. And one of the strategies that's been quite popular is to put some kind of implanted optical fiber into the brain and then to have a projection connector come out. And then you can actually buy off the internet, lasers that come with optical fibers ready to go, and you can then plug them in together. And then when you do that, the light delivery can be achieved quite simply. Now, if you really want to sort of discover how circuits work, though, then you need to be able to perturb more than a single point in the brain. And so that's something we've been working very hard on. In order to try and parse out how the complex three-dimensional circuits of the brain work together to implement things. And so one of the things that we've been working on is to try to develop massively parallel light delivery devices that can deliver light into complex patterns in the brain. So this is an array of optical fibers, each connected to its own miniature light source. The whole thing is about a cubic centimeter or so. And this one has 14 fibers which can tile the hippocampus in the brain bilaterally, which is one of the circuits that's thought to be involved with the formation of memories. And so it would be very interesting if we can delete information from the circuit just momentarily to see what is it actually storing at the beginning, end, or even after in a learning task in order to see what those cells contribute. And then, of course, we can label subpopulations of cells within this subpart of the brain so that we could try to label certain pathways or certain interneuron types or even certain neuromodulators that are still just being characterized. And so a big theme of this is that we have to really be sort of hyper-disciplinary about this kind of stuff. The molecules that we use to activate neurons, which come from algae and other molecules, have to be found through genomic mining. We collaborate with people all over the world, actually, to collect samples of different organisms, algae and bacteria and archaea and even funguses nowadays, in order to try and mine out these different light-sensitive proteins so that we can dial information in with the right level of precision and with the ability to address the right amount of the brain as well. And then, the next day we have to go back and then actually build these devices that allow us to get information into the brain. So one of the nice things of being at the Media Lab is that you're not really constrained by discipline. And so if you want to go in and devise circuit strategies to have arrays of light emitters that then can beam light into deep targets, you can do that. So one of the things we're very interested in doing is being able to sort of screen through the brain in order to go through the brain bit by bit and delete information. And the reason that's important is we've probably all heard about classical experiments where people would go through the brain and try to lesion information in the brain by just lesioning the brain itself. And part of the problem was that memories were actually found to be distributed. And so one of the ideas that we're trying to confront is by we back out entire set of circuits in the brain, and then if something happens, we'll subdivide until we hone in on exactly where that information is stored at a given time. Or we might find that there's a lot of redundancy. If we delete different parts of a circuit, it could be such that when we delete one part, other parts can take over. In many cases, in the clinical literature, the injury to a brain can result in some kind of plasticity that then can restore some degree of function over time. And so we need to be able to deal with a case where there might actually be very rapid adaptation to a brain perturbation. We also been working on ways to try and do this sort of high throughput circuit perturbations across brains as well. And so one of our former students, who's just actually spun out a company to commercialize these things, is making little couple gram wireless devices that are wirelessly powered and also can wirelessly control these light sources, such that you can put it onto, in this case, a mouse, but you can put it onto other critters, control many mice from one computer, and then you could do sort of high throughput screening of brain targets to try and really hone in on what are the causal substrates for a given action or behavior or decision. And so it's a very modular device, which has a little optics device here that goes to beam light into the brain. This is very similar to the one I just showed you. And then there's various computational power and communications modules there. And so this actually works pretty well. So here's just a quick demo. We're using a transgenic mouse, which is engineered to express the blue light activator that I just told you about pretty much throughout the cortex, and from Guoping Feng's group, also at MIT. When you drive these neurons with light, you can actually get all sorts of different effects. In this case, what we've done is we put this little wireless device over the motor cortex of the mouse, and when nothing is happening with the light, then nothing is happening with the animal. When we turn the light on, using a completely wireless interface, and the animal will start moving around. And so this is just a proof of concept that we can actually do the basic operations that we need to do these kinds of high throughput circuit screens. So another thing we've been working very hard on is how to turn off neurons, and that's important because the vast majority of neurons in the brain are off more than they're on. And so if you can delete information at specific points of time, that might be of great use. It's also the case that if you are driving all the neurons with a similar pattern, that might be a very unnaturalistic pattern. You actually would like to really dial information into individual cells or even subcompartments of cells. And so we've been working on finding molecules that are light-driven but can shut down neural activity. They do the opposite of the blue light activator from algae that I told you about before. So this genomic search really was inspired by this paper from, wow, almost 12 years ago, where they were looking at molecules called halorhodopsins, which when you shine light on them will pump negatively charged ions like chloride into cells. So that's the opposite of the positive charge in that we talked about before. Now these come from archaea, a very old class of organism, as it were, and specifically from halophiles that live in very high chloride concentration environments. And so brains, unfortunately, for this molecule, have chloride concentrations around here, and so the current, which is the y-axis, won't be very good. And one of these molecules, though, is different from the rest. For whatever bizarre evolutionary reason, its peak currents were splat dab in the middle of the brain chloride concentration range. And so a couple years ago, we published a proof of concept paper showing that indeed you could silence neural activity using this molecule, but the effects were somewhat weak. So we decided to try and cast the net more widely in this genomic search, and we set up a whole screening apparatus and system, and recruited people to really try and go through all these molecules from funguses and bacteria and all sorts of things to look for good neural silencers. So these are all from different species, and the molecules have different functions. Some of them are no good, as you can see, and then some of them are quite good. So last year we had one of our major hits, and this is a molecule that we nicknamed Arch. And Arch expresses really nicely in neurons and can also mediate pretty strong silencing currents, about an order of magnitude larger than the old one, both at low light powers and also at high light powers. So what that means is in the awake behaving brain, in this case an awake mouse brain, you can actually turn off neural activity 100% in the cerebral cortex. And you can do it in a very temporally precise way. In contrast to lesions, which might, well, they would delete things forever, I guess, or pharmacology, which delete things on the timescale of minutes to hours. This, you can see here, is shutting down a cortical module for about 5 seconds, and the latency to shutdown is very short, and then when the light turns off, the neurons start to fire again. Each of these dots is a little electrical potential being fired by a neuron. So now you can do all sorts of things, and I'll just tell you one short story about something we're trying to do, which is to understand how stress affects the brain. Now, stress, of course, is very commonly involved with all sorts of things, including psychiatric illness. It's something that contributes to depression. But also stress has evolutionarily important things as well. It can galvanize people to change behavior and to act and things like that as well. And so one of the questions is, how is stress actually modulating neural circuits to change how the information is being processed? And ideally, of course, we would do all these perturbations while we're recording from the target, so we know how the information is changing. But this is why I'm telling this the story in reverse order, as I alluded to in the beginning. The perturbation is a fairly simple and straightforward experiment, and we're still working on the readout methods. So in this case, we decided to look at another class of neurons. This is a collaboration with Kai Goosen's lab at MIT, which is to look at serotonergic neurons. And serotonergic neurons are neurons that you may have heard about in the context of topics such as antidepressants, fluoxetine, and other drugs being serotonin reuptake inhibitors, boosting serotonin's presence, at least so the pharmacology goes. And as you might have also heard, though, it can take many weeks for these SSRI drugs or serotonin boosting drugs to have an effect. And so a question is, how precisely do these serotonin neurons work? Are they really signaling information at a fast timescale, or are they really acting more like a volume knob on the circuit to turn up or down the functions of what's going on? And so we targeted serotonin neurons, again in a mouse, using this optical neural silencer, ARCH, and we were able to shut down these neurons pretty effectively. About 90%, 80-90% of the spiking could be deleted. And so we decided then to try and figure out if you delete serotonin just for a very brief amount of time, what happens? So we use a paradigm that Kai's group has used before, and many folks have used in psychiatry in animal models, because it's a way of assessing the effects of stress on the brain. And the basic idea is that if you have stressors, and there are many, many kinds of stressors, but we used a fairly brief form of stress, which is just an hour long on 2 days. And then you have some kind of learning paradigm. The stress will modulate the learning, and that's something that has been noticed. In this case, we're using a form of learning called Pavlovian conditioning. In this case, fear conditioning, where a tone ends with a mild shock, which is not painful, but it's annoying. And the fear conditioning results in animals fearing the tone that is being delivered. And so this is actually a useful learning paradigm that's been around in neuroscience for many, many decades. But it's also encapsulates some of the things that people think about when they think about stress. So, for example, recently there was a paper published about how people returning from war zones are more likely to return with post-traumatic stress disorder if they had prior stress before they went into the military. And so that's sort of a logical architecture of experiment that we're trying to capture here. Okay. So when you do this, you can see something like the following. So this is freezing, sort of, like the deer in the headlights response, in response to the tone. So animals are fearing the tone. And what you see is this is the blue is before the training and the red is after. So after the training of the fear conditioning, the animals fear the tone after more than before, as you might expect. With stress, however, the reaction is stronger, which emulates what we just talked about in terms of post-traumatic stress disorder being boosted by prior stress. And you can prove that the stress is actually modulating the learning, because if you put the fear conditioning before the stress, then there's no boost. Stress is not just generally ramping up emotional state, it's actually changing the learning itself. So now I decided to try and turn off these serotonergic neurons for just seconds in order to see how they contribute to the expression of stress in this learning modulation. And we decided to turn them off as opposed to on, and we decided to turn them off for a very brief amount of time rather than administering a drug for many weeks, because we really wanted to see in a very brief time scale what the causal role that these neurons could be playing in real-time brain operation was. And so we really are just deleting them for these brief a few tens of seconds during the tone exposure. And that's all the serotonergic deletion that we're doing during those two little time courses there. And interestingly, that was enough to completely block all the effects of stress, which is kind of interesting because, drugs like fluoxetine, the serotonin reuptake inhibitors would, so the theory goes, boost serotonin. Here we're finding that blocking serotonin is what's helping. And also, whereas those effects take many weeks, here we're finding the effects within tens of seconds. And so it really highlights how important it might be to really parse out how these different neural circuit elements work by deleting them and driving them at a time scale comparable to how the brain is actually computing, rather than purely looking at more gross effects over very long time scales. And this actually suggests new theories of how we might want to actually modulate this class of neurons in order to affect stress-related effects. Now, you might argue, well, we're turning off the neurons, but maybe it's a non-specific effect. You're just turning off the neurons for 60 seconds, and if you turn the neurons off for any 60-second period, 2 30-second periods, then maybe you would block these stress effects. And so if you look carefully at how we do the training here, we're using a very old style paradigm where some of these tones and shocks are paired so that they result in the fear of learning, but some of them are unpaired. They should have no information content, or if anything, should train the brain that the tone is not to be followed by a shock, depending upon how deep into the learning theory we want to go. So we did a different experiment. We silenced the neurons just during the unpaired tones. Now this would discriminate whether it's really all about the actual stimuli that are involved with the actual learning, or whether it's just modulating general tone of serotonergic neuron activity in the brain. More like the volume knob hypothesis again, rather than a very precise gate of sensory input into specific circuits. And when we did that, when we silenced these neurons for the same amount of time, but just shifted slightly so that we're not there during the actual stimuli that are leading to learning, then the blocking of these serotonin neurons has no effect. So you can really pinpoint the timing and the events outside of the actual circuit, in this case behavioral events, that are important for a given neuron class to gate information flow. And so this is something that we're very interested in following up. So we've continued to look for better tools. One of the things that we wanted to do is we collaborate with a number of groups that are trying to silence large brain regions, such as people who work with rhesus macaques, which are widely used in neuroscience to study cognition, perception, and other things that are more difficult to study in rodents. So one of the things we did was to do a genomic screen for molecules that were related to ARCH, since it looked like a pretty good molecule. And we found molecules that were a lot like ARCH, but were more light sensitive. And if it's more light sensitive, then you can actually try to perturb more of the brain. A single optical fiber of the kind we're using here might perturb, about a cubic millimeter or so of brain, maybe a little bit more. With this one, which is about 3.5 times more light sensitive, then you can perturb more of the brain. And so we collaborated with Bob Desimone's group at MIT, and they were able to show that they could actually delete activity in circuits in awake behaving rhesus macaques, which are species that are used to study attention and all sorts of other higher order forms of cognition that can be more difficult to study in other species. Now, one thing we've also been trying to do is to find tools that allow us to dial in information in more realistic ways. So far, we've all been talking about deleting activity or driving activity in sort of bulk tissue. And of course, that's a good first step, but you really want to be able to dial in information at a resolution of individual cells within the brain. And to point out how difficult this is, a cubic millimeter brain might have 100,000 different cells in it. So we're not there yet, but I'll show you in a second how we're trying to approach this problem, and we hope to be much better than what we can do now in a reasonably soon timescale. The second thing you can do is you can try to get different channels of information that you could use. So here what you can see is, once again, we have sort of efficacy on the y-axis, and now we have color on the x-axis. And one molecule has, you can see, pretty good activity in the blue, another molecule has activity in the red. And both of these are different silencers from different kingdoms of life. So if you put these two different silencers into two different neurons, one of them here you can see red light will quiet down, but blue light will not have any effect, or at least it'll be less of an effect. The crosstalk still exists to be up front. And that's what happens if you put this molecule in. If you take the more blue light sensitive molecule and put it in, then this neuron here will be quietable by blue light, but red light will not affect it as much. And so this was sort of the first demonstration that you could actually have two channels of deletion, where you could try to silence different neurons in a network and try to see how they work together, or whether information is handed off from one population to another, or other kinds of experiment like that. And so this is something that we've now distributed to many groups, and we're excited to push it further. And there's a lot of obviously molecular engineering things that you can do sort of in the synthetic biology space. So here's an example of just one last story before we go into kind of how to analyze what's happening in the brain, because that's very clearly becoming more and more of a addressable question if we can build the tools that allow us to confront those issues. This is from Akihiro Yamanaka's group, and what they were doing here was to label a very tiny cluster of neurons in the brainstem to make them silenceable with light. And these are neurons that atrophy or disappear in patients with narcolepsy. And so you might anticipate that if you turn off these cells, then you might be able to induce sleep. And so indeed, that's what they found. When you shine light into the brain, this is probability of being awake. When you shine light into the brain, these neurons turn off, and then the brain also goes into a sleep state within about 20 seconds. And it goes into a very specific sleep state. It goes into a state called slow wave sleep, which has been implicated in certain forms of memory consolidation and other kinds of interesting neurological and neuroscientific finding. You don't go into REM sleep, which is the state that's associated with dreaming and other kinds of phenomena different from the episodic memory consolidation. So it suggests that what's happening when you turn off these neurons is a very specific transition into sleep. It's not just a nondescript collapse of brain function, but it's rather putting the network into a very specific state. And you can even imagine how if you could put the brain into this slow wave sleep state, then it might actually be a useful thing to induce in the brain if you wanted to drive a certain kind of change in a memory state or other kind of phenomenon. So to summarize, we've distributed these reagents to about 500 groups worldwide. I didn't update this slide. We really try to collaborate with a very broad network of folks to get these reagents out into the world, nonprofit institutes to distribute the genes, and also viruses or transgenic animals that have the genes in them, so that it's more or less a turnkey technology nowadays, much less of an art form than it was about 5 years ago when our first papers came out. We also have a training program, and one of them is funded by the National Science Foundation to teach people how to do these kinds of experiments. So if anybody wants to learn how to do it and has a reasonable amount of prior experience on animal work, we can host you to learn how to do these things. But all is not clear, of course. We can perturb, but what is actually happening in the brain? What is happening to the computations? And as you might have noticed, we haven't talked a lot about actually what changes in neural coding or what changes in information transformation are going on. And part of it is that the tools for assessing brain information are also just to the point of now increasing in the scope where you could actually try to survey large fractions of a neural circuit. Here's an example of the kind of thing that we've noticed, and we published a couple papers that suggests why it's really important to survey what's happening in a circuit, even for the most simple interpretations of these kinds of experiments. All we're doing here is driving some excitatory neurons. This is just neural electrical potentials, and this is time, and there's a little blue bar here, which is the light. So neurons are firing now. And what you find actually is that a lot of neurons that you record from, we're recording with an electrode in the brain here, a lot of the other neurons actually are being quieted. So you might ask yourself, well, why is that happening? Well, the most likely explanation is that we're driving these excitatory cells. They're recruiting inhibitory cells nearby that then in turn quiet other cells. And then these cells are undergoing that quieting. And other groups are now investigating this by trying to perturb different inhibitory cells and finding that this is quite a plausible theory. Another interesting thing that you can do is if you quiet down cells, which is what's happening here, and these are all, again, electrical recordings of cortical neurons, then some cells will actually increase their activity. So in the beginning, we talked about, wow, this is better than lesions, because with lesions, the brain has time over many days to weeks to months to rewire. Here we're finding that even within milliseconds, look at the time scale here, there are neurons in the brain that are responding oppositely to the way that we're trying to control them. And so it highlights how, at an intellectual level, it's very nice to be able to record and understand what's happening to these neural codes, but at a pragmatic level, people really need to be doing this if only to understand what their perturbation has done at all. And so that's really what we've been trying to do. One of the things we've been working on is to try to develop electrode arrays and other microfabricated technologies that allow us to survey neural circuits in the vicinity of one of these optical probes. And what you see here is a close-up photomicrograph of a gold pad for recording neural activity, and then these pads are going all the way up and down this electrode. So when you put this probe into the brain, it looks like a finger, you can pick up neurons at multiple points along the axis of that probe. And it actually turned out to be harder to do this than we thought, because it turns out that the metal and silicon that people conventionally use to record from neurons is light sensitive. And so we actually had to work quite hard to look for materials like indium tin oxide, which is a transparent conductor, which avoid this artifact. But metals like tungsten and semiconductors like silicon actually respond to light with electrical potentials. And so here we have now a little gold pad then to pick up the spikes, and then we have this other pad which allows us to record in the frequency bands of activity where the artifact occurs. So now the question becomes, how do you actually tile a brain circuit in order to record these kinds of effects? And it's important to record locally, because that's where a lot of the strongest connections are, but it's also important to record very distantly, right? These three clumps of neurons I talked about so far, the dopamine neurons, the serotonin neurons, and these hypercretin orexin neurons, are all small, localized clumps of neurons that project very widely throughout the brain. And so if you want to understand how a reward impulse is changing information flow or computation in the brain to cause the brain to repeat an action or to make a decision in a certain way, then you might have to survey neurons very distant from where you are. And this is borne out by a lot of science that's been done. Groups have found that, for example, visual cortex neurons, just 2 synapses in from the retina, actually can encode for subtle properties of rewarding stimuli that have been experienced. And so